So now we're going to start our course. So first, we're going to discuss about the DNA preservation. So before we conduct any study, if you want to study the DNA, we need to collect the organism, and then we have to preserve the organism so that the DNA will be intact for the further analysis. So after this lecture, you should be able to first to know the procedure to collect biological samples for DNA study. So you have to know the procedure and also understand the principle and the method of the DNA preservation. So the procedure for the DNA preservation. So first you have to collect sample. After you collect the sample, you have to remove the tissue. So for example, if you want to get the DNA from an elephant, so you have to collect the elephant, okay? But you don't need the entire animal for the study. So you have to remove some of the tissue from the animal. And then after that, you have to document it. You have to document the sample. You have to label them properly and note down all the required information. After that, then you have to choose a method, preservation method to preserve the tissue and the DNA. So the objective is to first, you have to make sure you obtain the correct taxa, and you have to make sure you obtain enough DNA from organism, and you also want to minimize other compounds in the organism that could damage the DNA, and you want to reduce the contamination. So you only want the DNA from the organism or from the sample that you want to study, and you try to avoid the contamination of your sample by the DNA from other source. Okay, so see what we call the foreign DNA. So to collect the sample, so the sample is usually we refer to the organism, so it can be plant or animal. Okay. So first you have to determine the taxa for the research. Which species or which family or which genera that you want to study. So your research unit can be individual, species level, genera level, or family level. And you also have to decide how many samples you want to collect. And you want to collect the sample from the field or stock center. So the stock center, for example, is a nursery museum collection. So if you can find the samples there and the samples information like a location is available, then you can use the sample from the museum. So if you have to do the sampling, you can use the direct sampling method or indirect sampling method. So it's really depend on the research questions and also the organism. So the direct sampling method is what we call the invasive sampling. The indirect sampling method is what we call the non-invasive sampling. So for direct sampling, you have to find and collect the organism. So you have a direct contact with the organism. For the non-invasive sampling, you will not directly contact with the organism. So for the invasive sampling, you have to manipulate the organism. So you might catch, trap, or collect the organism. So you have learned all these things in the other course. However, many organisms cannot be collected directly. For example, protected species. So you're not allowed to harm the species, especially the protected species. Or maybe the organism is very hard to find, but somehow you still can get their DNA, or it's very difficult to catch. And sometimes you don't even know what is your target organism. So for example, in this case, those organisms that is very hard to find, see, and catch. But in any case, they might leave the trace of the DNA in the environment. Okay, so for example, you might get the DNA from their stool sample, okay, their fecal sample. So it's still possible even though you didn't see the animal. And sometimes you don't even know what is the target organism. So let's say you want to study the parasite in an animal. So in this case, you don't really know which parasite. So usually you just get the blood sample and hopefully extract the DNA from the blood sample. And if there's a parasite in the blood sample, probably you also can extract the DNA of the parasite together with the DNA of the host. So for the indirect sampling, non-invasive, you might collect the hair or fur of the animal. You might collect the feces, okay, and the parasite. So I give you a few examples. So I would like to show you one of the videos. So this video is a project named the Halimau Selamanya. It's a project conducted by RIMBA. So I play the video, so you just look at it and then try to observe and think what this tiger was doing. Okay. So what this tiger is doing? Okay, actually, what you see here, what is on the tree is a fur trap. Okay, so they put the pheromone, the hormone, on the trap, and when the tiger scratch their back on the tree, then the hair will trap on this device. And for the hair, so hopefully when the hair was detached from the animal, so this is the hair, hopefully there's a follicle where you can get some cells, and in the cell you can get the DNA of the tiger. So in this case, you don't need to trap the tiger, you don't have to have a direct contact with the tiger, but you still can get the DNA of the tiger. So this is another project by RIMBA. So for example, this is a flying foxes. Okay. So if you want to conduct a population study, you want to get the DNA from many individuals from a population. And in this case, if you want to get the DNA from these flying foxes, if you want to get the DNA directly from the animal, then you have to bring the animal down from the tree. And in this case, if you really want to do that, you might harm the animal. So one way to do it is to collect the feces, okay, and then get the DNA from the feces, because the feces has passed through the digestive system, the intestine and things like that. So there might be some cells left over in the feces sample. So another way to collect it is to collect the blood sample from the parasite or fly. Okay, so for example, this fly, they suck the blood from the animal. So what you can do is just get the flies and extract the blood sample from the flies, and you can also get the DNA of the mammal or animal that where the blood belong to. So after you collect the sample, so for example, if you use the direct sampling method, immersive sampling method, then you have to get the animal and then remove only part of the tissue because you don't really need the whole animal. So you might need only some of the tissue, so you can collect the tissue while the animal is alive or after is dead as soon as possible. Because after the animal die, the DNA of the tissue will degrade very fast. So you have to make sure you get the enough tissue so that you can obtain sufficient DNA. So for most of the DNA extraction protocol and the downstream lab procedure, you only need small amount of the tissue. So for example, this is one of the extraction kit, okay, extraction method. So this is the amount of the starting material, so the tissue that require for you to get the certain amount of the DNA. So you can see for different type of tissue, you need different amounts, and the quantity of the DNA also differ. So for small organism, usually we will use the entire animal or the whole plant. So for large organism, we will just take a piece of tissue. So you have to select the part of the plant or animal that are relatively free from the compound that can damage the DNA and the compound that can inhibit the subsequent molecular work, for example, the PCR, and also the foreign DNA. So for the plant, so to avoid those unwanted DNA and also the material that could damage the DNA. So we need to, for plant, usually we select a young or actively growing leaf. Okay, so for the plant, we usually take this part. But other parts are okay as well, but if possible, you try to choose the part that is young and actively growing. For animal, so you can use any type of tissue. For insect, okay, so usually you will take a piece of the leg, okay, here, this part, where inside the skeleton, there are a lot of the muscle tissue. So this part is rich in DNA. Or we can get the muscular tissue from the wing. And for the butterfly, you can get the DNA from the thorax or head. So there are few important things you have to remember when you remove the tissue. You have to make sure all the scalpels, forceps, vials, and container, all the material and apparatus that you use for when you try to cut a piece of tissue from the animal, okay, sterile. Okay. You also need to make sure you weigh all the curve so your DNA will not contaminate the sample. So that means that in your lab practical, you have to make sure you sterilize all the material or apparatus that you can sterilize. So the next thing, after you have collected the sample, remove the tissue that you need. The next thing you have to do, you have to document the detail of the sample. So this is very important. So you have to label your sample properly. So usually the proper label, you will have a voucher reference number. Okay. You will have a location information, who collected the specimen, when the specimen was collected, and also taxonomy information and the photo. So all this information is very important when you preserve and collect the sample. Okay. So after you have collected the sample, okay, then you have to keep the sample properly. Okay, usually you will keep in the collection or museum. So you have the voucher specimen. So this can be the leftover of the organism. So it's just for the organism, you just take part of the tissue. Then the remaining of the organism, you just keep it and put it a proper label. And after that, you have the physical specimen. You also need the specimen information. So specimen information is the information that I showed just now regarding to the taxonomy, location, collection type, and others. After you have these two, then the next thing you can do is to proceed to the molecular work to get the DNA sequence. Okay. After you complete your study, then all this information need to be integrated and cross-refer to each other. So we're going to discuss more of this during the DNA barcoding and also the sequence repository database. So we're going to discuss this in the future in more detail. So you just have to remember you have to collect all the data and keep it properly. So after that, okay, after you take a piece of tissue from an animal, okay, and then you already label them properly. So the next thing you can do is to preserve the tissue. You have to put the tissue in chemical or in a fridge to preserve the tissue and also the DNA. So remember, once you have removed the tissue, you have to preserve the tissue as soon as possible. So this is to prevent the DNA from degradation and also denaturation. So DNA is a molecule like many other molecules. DNA can degrade and denature easily. So just now we talked about the degradation and denaturation. So both are not good for DNA, but these are the different type of the damage on the DNA molecule. So you have to know what is the degradation, what is the denaturation, and what caused the degradation and what caused the denaturation. If you know the cause of the DNA degradation and denaturation, then you can do something or use any chemical to reduce the cause of that, reduce the agent or the cause. Okay. So you have learned this before, so I'm going through quickly. So this is a nucleotide. You know that the DNA is a double helix molecule, and each of the nucleotide is connected with the phosphate bond. Okay, and it's a pair. You have a double helix structure of a DNA, and each a pair of the strand of the DNA is attached to each other with the hydrogen bond. Okay. So for DNA degradation is referred to the fragmentation of DNA molecule into the nucleotide. Okay. So that means that this phosphate bond is disappear. Okay, it's damaged. Or the carbon-nitrogen bond, which is this one. Okay, this is a carbon, this is a nitrogen bond. Okay, for example, this one. Okay, so the main cause of this degradation is the oxidation effect, autolytic effect, a process. Okay, for example, some enzyme activity. So this will cause the DNA double helix structure, still with double helix structure, but it will break in the smaller fragment. So for denaturation, it will not affect the primary structure. Okay. So it's mainly refer to the breaking of the covalent bond, okay, this bond between both strands of DNA. Okay, so you can see the primary structure is still intact. So the cause is mainly due to the hydrolytic effect. Okay, so this is a H2O. Okay. So now we know the the cause of the DNA degradation and denaturation. So there's an oxidation effect, autolytic effect, and also the hydrolytic effect. So we can look for the way to minimize all this effect and activity. So one way to do it is to keep your sample, the tissue, in low temperature. Then you can lower the enzyme activity and exclude the light or other radiation. So in this way, you reduce the oxidation effect. You also can remove the water, then you can reduce the hydrolytic effect. Okay, so these are the things that you can do. So for oxidation, you also can remove the oxygen. Okay. At the same time, you also can sterilize your sample or the surface of your sample. Okay, reduce the microbial, so you reduce the DNNH activity. So we know that these are the principles, then we can choose the method. For example, to keep the sample in a low temperature, you can keep your sample in freezer in different temperature. If you want long-term storage, it's better to keep in a very cold and agent environment. To exclude the light, you can store the sample in a dark cabinet or room, or use the aluminum foil to wrap the glass bar. Okay. To remove the water, you can air dry the sample by using the food dryer, or you can use the silica gel, okay, to remove the water from the sample. And you also can use ethanol to replace the water in the tissue with the ethanol. To remove oxygen, you just store your sample in an airtight container. To sterilize against the microbial, so you have to make sure you wear the hand glove and sterilize all the apparatus, okay, by autoclave or by Bunsen burner. So these are the few simple things you can do. So when you want to preserve a sample, usually you use a combination of few of these methods. So that's all for the DNA preservation.